In my team, we quantify the interactions of molecular and cellular systems. We use this information to control the systems. Tetiana Serdiuk, in her work, characterized membrane receptors, how they receive extracellular signals and transfer these signals to the interior of the cell. Now, she particularly focused on the ß-adrenergic receptor, which is important for the heart diseases and for lung diseases, and she quantified how these receptors interact with cholesterol, a major component of the cell membrane.
I would like to show you our latest study where we revealed very unexpected role of cholesterol in stabilizing G-protein couplet receptors, or GPCRs. We used as a model system ß-adrenergic receptor. This is one of the most studied members of this family, and it's used in the field as a model. It's also a very important G-protein couplet receptor because it senses adrenaline and noradrenaline, and it is very important drug target. We were actually specifically interested to understand how cholesterol stabilizes this receptor. Why it's important? Because we know in biology that the structure of the protein, this is what you see here, this arrangement of helices and loops defines the function of the protein. And if protein loses this structure and it gets destabilized, it unfolds, it's no longer able to perform its function. Therefore, it's really crucial to understand how the structure is stabilized. And cholesterol, which you see here in green, we found that it binds to this receptor in the membrane, and we specifically asked how does it interact and stabilize this receptor at different temperatures, at 25 degrees, which is room temperature, at 37 degrees Celsius, which is our body temperature, and also at hypothermia, which is 42 degrees. This is the condition when you have fever, which starts to damage your body. And what we found, surprisingly, that cholesterol only slightly stabilizes this receptor at 25 degrees, but it strongly stabilizes it at 37 degrees, and it loses its stabilization effect at 42 degrees. It's no longer able to stabilize the receptor at this temperature. Our research shows that it's very important to study these receptors not at the temperature which we have in the lab, so room temperature, which is actually regularly done today, but actually at the body temperature, physiological temperature of 37 degrees.